rough strain and smooth strain and injected it into the mice, because it take mice. Why it take mice? Because the anatomy and physiology of a mice are very similar to the human beings. That is, it is one of the experimental material which is used in the laboratory to testing different types of medicines and different types of result. So rat are taken here. What happened? The rough strain, when injected into the mice by injected into the mice, and because it is non-virulent in nature, so the result will be obtained here. The mice is lives. It means that might cannot that take place. That cannot be take place of the mice. But when smooth strain of Diplococcus pneumoniae are taken and injected into the mice is dies, because it is virulent in nature. So mice is died. Then after that, what happened? When Griffith heat killed the smooth strain, when he heat killed the smooth strain, and this heat killed smooth strain are injected into the mice is live. And in his fourth experiment, what he can done, he taken rough strain, this is non-virulent strain, rough strain, and heat killed smooth strain. Heat killed smooth strain is non-virulent in nature, as you seen in this experiment, third experiment. And in fourth experiment, when he taken a mixture of rough strain and heat killed smooth strain and injected into the mice will die. Mice will die. So here, what conclusions are coming? When the heat killed smooth strain and rough strains are taken together, injected into the mice will die. So Griffith says that something is from the smooth strain of the Diplococcus pneumoniae passes into the rough strain, and this rough strain passes into the rough strain, and this rough strain show the nature of virulency in the mice. So the mice is die. But after performing such type of experiment by the Griffith, he does not find out what type of a molecule that is transferred from the heat killed smooth strain to the rough strain. He cannot find out it. After Griffith, three scientists, Avery, MacLeod and McCarty, proved that the substance that is responsible to the dies of a mice is a DNA, and it is transferred from one generation to the another generation of the mice. What is the result coming here? Heat killed smooth mice, when injected into mice, the mice will live. Why this happened? What is the reason that heat killed smooth strain is so non-virulence in nature, and rough strain plus heat killed smooth strain so virulence in nature? What happened? Actually, when the smooth strain of Diplococcus pneumoniae, it having a covering around it, and this covering is made up of polysaccharide, when heating should be take place, the polysaccharide covering of such a strain is destroyed, and it can be seen here. What happened? The heat killed smooth strain does not having the covering of polysaccharide. That's why it is non-virulent in nature. But what happened? When the heat killed smooth strain is mixed with rough strand, then this is rough strand and this is smooth strand. Something is passes from it does not having this heat killed smooth strand does not having the polysaccharide covering around it. Then what happened when these two things are mixed together? The DNA that is present in the smooth strand is passes into the rough strand, and the DNA passes into the rough strand, and this DNA is responsible to develop this DNA having a gene responsible to develop a capsule around it. So this capsulated and it become capsulated bacteria. Become capsulated bacteria, and which is virulent in nature. Something is passes from heat killed smooth strand to the rough strand, and the rough strand become virulent. That's why the mice will die. With this experiment, it was proved that DNA is a genetic material, but Griffith does not explain it because he does not know about the DNA molecule. Avery, McLeod and McCarthy, these three scientists proved that the substance in the Griffith experiment was DNA. So, this is all over the Griffith experiment, and this Griffith experiment is called transformation. Transformation. So what is transformation? Transfer of DNA segment from the surrounding medium, donor medium, to the acceptor. So what is transformation? Transformation is a transfer of DNA segment from the surrounding medium, donor, to the acceptor. Thank you. This is all over the Griffith experiment.